My name is Bob MacArthur. I'm a behavioral pharmacologist. Most of my professional life has been spent in the discovery and development of drugs used in the treatment of psychiatric and neurological disorders. My work has been concerned with depression, anxiety, alcoholism, Alzheimer's, Parkinson's, and epilepsy. Professionally, behavioral pharmacologists work with animal models of these and other CNS or central nervous system disorders. We alter behavior in animals so that they resemble the clinical symptoms of the disorder in humans. We then try to improve these behaviors using drugs that are effective in the clinic and then compare them to new molecules that we have theoretical reasons to believe would also be effective in their treatment. We work to provide a profile of the beneficial activity of these new molecules in animals that hopefully will translate into drugs. My lab has been associated with the successful launch of clinically effective drugs. However, the number of successful drugs is low, and most compounds that preclinical drug discoverers work on unfortunately never pass the clinical phase 3 golden standard of clinical efficacy. The focus of my work for the last 8 years has been concerned with trying to understand the reasons why there's such a low translatability between preclinical and clinical efficacy. And more to the point, what can be done to improve this translation? To further this understanding, I have recently published a number of articles and books examining the relevance of animal models to CNS drug discovery. You know, I'm often asked this question, not only by young scientists, but also by experienced project leaders and department heads. They ask me, if I had to choose one animal model, which would be best to predict clinical activity? The short answer is none. Why? Well, first, the causes of psychiatric disorders such as depression are very poorly understood. Secondly, behavioral disorders are not exclusive to any one clinical category. Even neurological disorders such as Alzheimer's, where the actual physical changes in the brain can be seen and measured, the relationship between the physical changes and behavioral manifestations, such as cognition in Alzheimer's disease, for example, is not clear. We cannot capture all the manifestations of a particular neuropsychiatric disorder in one animal. The best we can do is model aspects or phenotypes of abnormal behavior to try and understand the neurobiology underlying such abnormalities. So in many ways, it's actually a nonsense to talk about an animal model of a disorder. And perhaps we should be talking about animal models for CNS disorders. Let me be quite clear. I'm not saying that animal models for CNS disorders are irrelevant because they need not necessarily predict clinical efficacy. They are extremely relevant for the study of the neurobiology of neuropsychiatric disorders of which we understand so little. And in so doing, they help identify and validate the drug targets that the biopharmaceutical industry are working on. As such, animal models are essential to the process of drug discovery and development, not only for CNS disorders, but all other pathologies. By modeling aspects or phenotypes that can be shared across a spectrum of CNS disorders, we gain a better understanding of the neurobiology of neuropsychiatric disorders, and this understanding helps discover and develop new chemical entities that can alter the expression of these phenotypes behaviorally, physiologically, and molecularly. Therefore, not only do we gain greater insights of the causes of the disorders, we can also provide new drugs for their amelioration. Will you improve your chances of developing and registering a new drug for the treatment of neuropsychiatric disorders? Well, you'll certainly improve your chances, but no one can guarantee that regulatory bodies will accept a candidate drug as a clinically effective pharmacological treatment. Registration of a new chemical entity as a pharmacological treatment is dependent upon demonstration of clinical effectiveness in a phase 3 trial. Although earlier phase clinical trials, which may have relevant clinical endpoints built into them, may show promise of clinical effectiveness, if clinical improvements is not demonstrated in the large phase 3 trials, it's unlikely to be approved. First of all, behavioral changes in neuropsychiatric clinical trials are measured using clinical rating scales. These include, say, the HAM-D for depression, PANS schizophrenia, MMSE or ADS-COG in Alzheimer's disease. They are intended to tap into the clinical benefit of the treatment in patients such as feeling less depressed, less anxious, or able to remember better and take care of themselves longer in the case of Alzheimer's patients. As I indicated earlier, improvements in animal studies are based on behavioral responses of animals that have been deliberately impaired. In many ways, we are faced with an apple-orange comparison. We infer that impaired behaviors in animals reflect abnormal behaviors in humans. We assume, by observing improved behavior following pharmacological treatment in animals, that a similar improvement will manifest themselves in patients. They may be, but other factors characteristic in neuropsychiatric clinical trials may override a clear improvement in clinical symptoms. A great problem in neuropsychiatric clinical trials presently, for example, is the placebo effect. This is an effect where patients are being treated with a sugar pill or an inert substance will get better without further pharmacological treatment. The placebo effect can produce failed trials where no treatment condition is significantly different from placebo. So sometimes a clinically active comparator fails to show a clinically effective improvement. Secondly, human clinical trials, especially large-scale phase 3 trials, have their own issues and problems. Aside from the subjective nature of clinical rating scales and their interpretation, and shifting baselines through placebo-related mechanisms, as more and more large-scale clinical trials are being done in different countries, issues of language and culture, they will all contribute to the complexity of using such scales. Many other problems exist with the design and conductance of neuropsychiatric clinical trials that are not the topic of the present interview. Yes, there are. There's a great emphasis on developing experimental medicine or translational research teams throughout the industry, government, and academia. These teams consist of both preclinical scientists working with animal models and the clinical investigators that are ultimately charged with the safety and conductance of human experimental medicine studies. They are actively engaged in establishing biomarkers that hopefully will give us a handle for the diagnosis and subsequent treatment of neuropsychiatric disorders. These biomarkers are physical, biochemical, physiological changes that occur in the body during the course of a disease and disorder. By identifying similar markers in animal models, all the disorders, the results of behavioral studies in both, will be further strengthened. For example, neuroimaging in humans and small animals are identifying comparable changes in human patients and in animal models of CNS disorders. These can be used as cross-species points of reference. Similarly, cross-species neurocognitive techniques are being developed that can tap into similar processes in men and animals, thereby providing a common measure in both. As more cross-species congruent responses, as say for example genetic, imaging, biomarkers, behavior, etc., are identified and exploited, they can provide more objective measures of efficacy throughout the drug discovery and development process. These will strengthen the evidence of clinical improvement, supplementing those based on subjective clinical rating scales. Thank you for your time.